Testing, one, two, three. Can everybody hear me? Please give me a thumbs up if everyone online can hear me. Great. It's 1 p.m. Good afternoon to everyone. I trust everyone is well. Welcome to today's webinar titled Differentiating Between Stalk Borer and Armyworm Species. While the session is scheduled for 3 hours, I suspect it will likely end before that. CropLife SA members registered on the CPD system can use the extra time to complete the very easy assessment loaded in category 1 on the CPD platform. A good deal in my view. I do apologize if I drop off for a minute or two given the load shedding in my area. Let's hope not. Fingers crossed. So in terms of the content, we will focus on 3 stalk borer species and 2 armyworm species. While other hosts will be briefly mentioned, the focus crop will be maize, given the relevance of the pests to be covered in the context of maize. This webinar will start with a brief introduction on the 5 species. Under identification, the eggs, larvae, pupae, and moths will be covered for each species. The life history of each species and feeding damage will be discussed. We will then recap the key points and differentiate between the stalk borer species, armyworm species, and between stalk borers and armyworms. The importance of detection and monitoring will be looked into in terms of the pests covered. Management strategies will be discussed. This will largely involve cultural methods, insecticides, and genetically modified maize. The advantages of record keeping will be touched on using brief examples, and finally concluding remarks will be made. So without wasting any more time, let's get straight into it. Okay, so now surveys done in the past have shown that there are more than 130 species of stalk borers present in East and Southern Africa, with 20 of these species that are of economic importance. Bassiola fusca, commonly referred to as the African maize stalk borer, described and named by Fuller. Chylopatelis, commonly referred to as the spotted stalk borer, described and named by Swinow. And finally, Sesamia calamistis, commonly referred to as the pink stalk borer, described and named by Hampson, are considered the most important stalk borer species in Southern Africa on maize and sorghum. All 3 stalk borer species belong to the insect order Lepidoptera. Bassiola and Sesamia belong to the moth family Noctuidae, whereas Kylo belongs to the moth family Crambidae. And yes, everyone, stalk borers are indeed moths and not worms. And what all of you see on your screen is the immature life stage known as the larval stage of development. It is easy to understand why many people refer to the larval stage of lepidopteran pests as worms, because as all of you can see, to someone who has no entomological field experience, those images look like worms. For all of you here today, this stage will no longer be referred to as worms, but rather as larvae. Now, Bassiola fusca and Sesamia calamistis were present in South Africa before the extensive cultivation of maize, sorghum, and other crops began. After the introduction of extensive cropping systems, these species became pests and are now considered to be important pests of maize and sorghum in the country. As early as 1920, annual maize yield losses of up to 10% was ascribed to Bassiola fusca, making this species the major pest of maize in South Africa. It is estimated that second-generation larvae of the species can cause up to 100% yield loss if left unchecked. Similar to Bassiola fusca, Chylopatelis is also important in the context of cereal crops. Of the 3 stalk borer species, Chylopatelis is exotic to Africa and originates from Asia. Chylopatelis can be found in mixed populations with Bassiola fusca in maize. Although Chylopatelis is not indigenous to Africa, it is still one of the most damaging species of stalk borers that occurs in this region. Male yield losses in East Africa attributable to Chilopartelis infestation are estimated at 15 to 45%. In South Africa, yield losses caused by Chilopartelis in maize have exceeded 50%. Sesamia calamistus, while considered an important stalk borer species, it is generally less important when compared to Basiolus fusca and Chilopartelis as a pest of cereal crops in East and Southeast Africa, but may be locally abundant. It is only a serious pest of cultivated cereals in Central and West Africa, where yield losses of up to 100% have been recorded where the species was found in mixed populations with Basiolus fusca. In southern Africa, the highest infestation levels are generally found late in the season. In the Eastern Cape province of South Africa, Sesamia calamistus infestation levels of up to 75%, with as much as 13 larvae per plant has been reported. Surveys conducted in other African countries have shown that losses associated with Sesamia calamistus account for less than 5% of potential harvest. Secondary crop hosts are pearl millet and sugarcane for Basiolus fusca, and in the case of Chilopartelis, these include pearl millet, rice, and sugarcane. Now, all three stalk borer species are not limited to only primary or secondary crop hosts, but can reproduce on wild grass plant species, typically belonging to the Poaceae, Cyperaceae, and Typhaceae families. The wild host species serve as a reservoir where borers survive when crops are not available. There are more than 30 different wild host grass species in Africa that can be infested by stalk borers, where some of the wild host species have been found to have higher infestation levels than crops. This could be due to various reasons, including the fact that some of these wild host species do not consist of thick enough stems for the larvae to penetrate. An example of such a species is Napier vetiver, which can be used together with Desmodium in a push-pull system for the control of stalk borer pests. This will be discussed further under management strategies. Fall armyworm, known as Spodoptera frugiperda, indigenous to the tropical regions of the Western Hemisphere. As all of you would know, or many of you on this online today would know, is a polyphagous migratory insect pest that feeds on more than 80 plant species, which includes maize, rice, sorghum, and sugarcane, but also vegetable crops and cotton. It has a voracious appetite, reproduces and spreads quickly given optimal environmental conditions. In South Africa, this migratory pest invaded during the 2016-2017 maize production season and has shown clear preference for maize followed by sorghum crops. This pest has since been recorded in all 9 provinces of the country. It is important to note that the fall armyworm is a regulated pest in terms of the Agricultural Pests Act 1983, Act number 36 of 1983, under the control measure R449. It still remains mandatory to report the presence of this pest on farms to the Department of Agriculture, Land Reform and Rural Development, better known as DLARD. Now, while the African armyworm is a migratory species like the fall armyworm, it is indigenous to the African continent. Often when it comes to this species, what many notice are large areas of lawn, pastures, or grass crops, such as maize and sorghum covered in dense colonies of larvae. This will be expanded upon when we discuss the species' life history. In general, the African armyworm is distributed widely in sub-Saharan Africa and tends to be a serious pest in countries north of South Africa. Let's move on to identification. The eggs of the African maize stalk borer are creamy white when laid, but darken just prior to emergence. They are approximately 0.8 to 1 millimeter in diameter, hemispherical, and slightly flattened with about 70 crenulations, also referred to as ridges on the egg shells. The larvae, yes, larvae, not worms, of the African maize stalk borer lack conspicuous hairs or markings. They are usually creamy white in color, often with the distinctive gray tinge, but also sometimes with the pink suffusion. The head capsule is dark brown, and the prothorax is yellowish brown. The spiracles, breathing holes found along the side of the body, as you can see here, are elongate oval with black edges. The larvae have prolegs along the abdomen, as you can see here. The pupae of the African maize stalk borer are dark brown and approximately 25 millimeters long. Parts of the African stalk borer have a wingspan of 25 to 40 millimeters. Forewings are light to dark brown with dark markings. As everyone can see quite clearly here. Hindwings are white to gray brown, as we can see here. May not be too clear, but we can sort of see the white and then going to gray brown there. I think it becomes very clear that both male and female moths are quite similar in appearance, and that I think is quite clear on all of your screens as well. Okay, so moving on to the spotted stalk borer. So eggs of this species tend to be flat, oval, creamy white, and approximately 0.8 millimeters long. Okay, spotted stalk borer larvae are creamy white to yellowish brown in color. They have 4 purple-brown longitudinal stripes, usually with very dark brown spots, as everyone can see, along the back. This gives the larvae a spotted appearance, and I think that's quite clear. When fully grown, larvae have a prominent reddish brown heads, and I think that's also quite clear on all of your screens. Okay, the pupae of the spotted stalk borer are approximately 15 millimeters long, slender, shiny, and light yellow-brown to dark red-brown in color. Spotted stalk borer moths are relatively small moths with the wingspan of about 20 to 25 millimeters. Forewings are generally brown yellowish with the darker scale patterns forming longitudinal stripes. I think that's the best picture I could come up to depict that. In males, hindwings are pale straw color. In female, hindwings tend to be white. Okay, I couldn't quite find one with the females showing the hindwings, but I think with the males here, we can also see that sort of pale straw color that comes through. And I think, yeah, that comes with some entomological training as well to spot those differences. Okay, so eggs of the pink stalk borer are about 1 millimeter in diameter, hemispherical, and slightly flattened with radial ridges. They are creamy white when laid, but darken as they develop. Pink stalk borer larvae look smooth and shiny and lack obvious hairs or markings. Larval color varies, but is usually creamy white with a distinctive pink suffusion. The head and prothoracic shield, that's the head there, that's the prothoracic shield there, are brown. The dorsal part of the last abdominal segment, as we see here, bearing the anus, is yellow-brown. Spiracles are elongate oval with black surrounds. Mature larvae are about 30 to 40 millimeters long. The pupae of the pink stalk borer are around 18 millimeters long and brownish to yellowish brown with the wrinkled frontal region of the front head, as we see here. The wingspan in female pink stalk borer moths is about 20 to 30 millimeters, and in males a little less. The forewings are pale brownish with variable but generally inconspicuous darker markings along the margin. And overall, I think what's quite clear is the silky appearance that comes about quite clearly. Hindwings are pearly white with the yellowish margin, which unfortunately I can't show all of you because I could not find an appropriate image there. Okay, so fall armyworm eggs are dome-shaped and are gray-white in color. Eggs are covered in protective scales rubbed off from the female moth's abdomen after laying, giving the eggs a hairy or moldy appearance quite clearly there. Okay, so the young, small fall armyworm larvae are usually light green to brown with a dark head capsule. Mature large fall armyworm larvae grow to about 3 to 4 centimeters, becoming darker as they mature, with pale white stripes along the length of their body. The large caterpillars have a pale inverted Y shape between the eyes, as pointed out there. 2 dark spots with dark spines occur on each body segment. On the upper body surface with 4 black spots. Again, quite clearly pointed out here, arranged in a square on the second to last body segment of the larvae, of the mature larvae. So the pupae of the fall armyworm are shiny brown and between 1.3 and 1.7 centimeters long. Fall armyworm moths measure 3 to 4 centimeters from wingtip to wingtip. Female moths are slightly larger than males, although both sexes have a white hindwing with a dark brown margin. Again, clearly, I think this picture shows that quite clearly here, the dark brown margin. Male moths are more patterned there and have distinct triangular white spots at the tip, but also at the center of each forewing. And that's really what's used morphologically to differentiate between male and female fall armyworm moths. Okay, the eggs of Spodoptera exempta are pale yellowish, darkening through development until just before hatching. The black head capsules of the larvae can be seen through the shells. Each egg is about 0.5 millimeters in diameter, conical with a slightly rounded apex and a densely sculpted surface. Eggs laid are generally covered by blackish scale hairs from the tip of the female's abdomen, and this covering may be sparse on later batches. Okay, the larvae of the African armyworm grows to about 25 millimeters in length. In the swarming phase, African armyworm larvae are usually blackish in color, but brown to green otherwise. Similar to fall armyworm larvae, they also have an inverted Y marking, as pointed out there in the head region, as well as similar lateral stripes and bands. However, they lack clear spots on the upper body surface, and I think that's clear to see here compared to what we saw with the fall armyworm in the previous slides. Okay, so Spodoptera exempta pupae are mahogany brown in color, 10 to 14 millimeters long with a smooth shiny surface. Now, generally speaking, it is difficult to distinguish pupae of Spodoptera species. I can take it one step further here. In general, for someone without entomological training, it would be extremely difficult to distinguish between the lepidopteran pest species we are discussing today based on the pupae. I would like to think that this is something all of you have picked up on, and during this part of the webinar, and as we move along, all of you will start to see how the puzzle comes together. Okay, so Spodoptera exempta moths are stout-bodied moths of typical noctuid appearance, about 14 to 18 millimeters long and with around 29 to 32 millimeters in terms of wingspan. The abdomen is covered with pale gray-brown scales, except for the tip in the female, which has black hair scales characteristic for the species. Forewings are dark brown with distinctive gray-black markings, as can be seen on your screens. Hindwings are white with dark veins. In essence, African armyworm moths look similar to their fall armyworm counterparts. And with that, we are now done with identification and we'll be moving on to life history. This is the point where all of you need to start sipping your coffee and concentrate. We are now going to go into more detail and start putting the puzzle together. To be clear, maize will be the focus crop as noted at the start of the webinar. Okay, so Bassiola fusca occurs in most agro-ecosystems throughout South Africa. These habitats where this species can be found includes areas with relatively low altitudes, coastal areas, and areas that are in the mountain areas of altitudes of up to 2,130 meters above sea level. Flight patterns of Bassiola fusca differ between regions. This could be due to differences in climate in different areas. There are 3 moth flights in the warmer central part of South Africa, where flights are spaced approximately 9 weeks apart. The first moth flight in Potchefstroom in the Northwest Province commonly takes place between the months of October and December, followed by the second flight during January and February, and the third flight occurring during late February until mid-May. The 3 flights can be distinguished from one another by a drastic decline in moths between flights, although there are seldom periods of time where no moths are present after the second flight. Female Bassiola fusca moths lay eggs in a row between the stem and leaf sheath. Each female lays on average about 200 eggs over its short lifespan of several days. Its exact duration depends on temperature and other factors. Egg laying on maize is usually concentrated on plants that are less than 2 months old. The leaf sheath of the youngest unfolded leaf is the most attractive part for females to deposit eggs. The eggs hatch in around 7 to 9 days and larvae move into the leaf walls to feed. Third instar larvae tunnel into stems where they feed for about 27 to 29 days before pupating within the stem tunnels. Now, very quickly, what do we mean by larval instars? And no, we are not talking about stars you see in the sky. An instar is the name given to the developmental stage of an arthropod, in this case an insect between molts. For example, after hatching from the egg, the larvae is said to be in its first instar. When the larva molts, it is then a second instar, and so on. For certain noctuid pest species, there can be up to 9 larval instars prior to pupation. Bassiola fusca moths emerge after 9 to 11 days from a hole that was produced prior to pupation, commonly referred to as an emergence hole, as pointed out on your screens. These moths live 5 to 9 days and eggs are laid 2 to 4 days after emergence. Under favorable conditions, temperatures of 25 to 30 degrees Celsius and 50 to 60% relative humidity, the life cycle can be completed in 7 to 8 weeks, but during dry and or cold weather conditions, the larvae can enter a period of suspended development known as diapause of 6 months or more in stems and other plant residues until conditions improve and become favorable for development. For example, when the rains arrive. This is evident during the winter months in the summer growing regions in particular. Now, before I move on, all of you studious souls would have figured out that developmental time from egg to adult is variable depending on environmental conditions, in particular temperature. And the question now becomes why? The answer is simple. The insect pests covered in this webinar are exothermic, meaning cold-blooded, where their body temperature and growth are affected by their surrounding environmental temperature. These insects require a consistent amount of heat accumulation to reach certain life stages such as egg hatch, larvae to pupae, or adult flight. Therefore, developmental time from egg to adult will vary between regions depending on environmental conditions, in particular temperature. The spotted stalk borer does not occur in the cooler eastern and southern parts of the high veld, but thrives in warmer parts in the northwestern areas of South Africa. This species also thrives in more coastal areas of the KwaZulu-Natal province and the lower parts of the Mpumalanga and Gauteng provinces. There are 2 distinct moth flights during spring and in late summer. The first peak occurs during September to October, and the second from February until the beginning of May. Eggs are laid by female moths in overlapping batches of 10 to 80 eggs on the upper and underside surfaces of leaves that hatch after 4 to 10 days. Larvae begin feeding in the leaf wall. The older larvae then tunnel into stems where they feed extensively and grow for 2 to 3 weeks. When larvae are fully grown, they pupate and remain inside the stem as pointed out on your screens. Moths emerge after 7 to 14 days and live for 3 to 8 days, during which time they mate and lay eggs again. During the dry, cold periods, typically characterized by the winter months in the summer growing regions, and like Bassiola fusca, larvae of Chilopatelis generally enter a state of suspended development. Again, the term used to refer to this is diapause, which lasts for several months. Pupation will only take place when the rains arrive. In general, moth emergence occurs in the late afternoon or early evening. As with all species covered in this webinar, the moths are nocturnal, meaning that they are active predominantly at night. During the day, they tend to rest on plants and plant debris. The whole life cycle for Chilopatelis takes approximately 3 to 4 weeks, which varies in relation to temperature and other factors. Under favorable conditions, 20 to 30 degrees Celsius and 60 to 80% relative humidity levels, 5 or more successive generations may develop. Now, in South Africa, Sesamia calamistus occurs along the coastal regions, the low veld of Limpopo and Mpumalanga, northwest, and parts of the Maize Triangle. The pest mainly occurs in coastal regions where it is considered important, but inland infestations have been on the increase as well. Similar to the Borer, pink stalk borer moths start flights as early as September and can have up to 5 overlapping generations per year under favorable environmental conditions. In summer rainfall areas, infestations are more severe during October, November in seedlings, and in February, March when cobs are present. Female moths lay their eggs between the lower leaf sheath and the stem in batches of 10 to 40 eggs arranged in 2 to 4 contiguous rows. On average, each female lays around 300 eggs in 5 days. Eggs hatch in 6 to 9 days. Ingram in 1958 reported first instar Sesamia calamistus larvae, as pointed out on your screens, to feed for approximately 1 week in the wall of the plant, after which they penetrate the stem. However, van den Berg and van Wyk reported the vast majority of first instar larvae to feed on the leaf sheath before directly penetrating the stem of the maize plant. Larvae remain inside the stem or ears of the maize plant until they pupate. Development of the larvae takes 4 to 6 weeks. Sesamia calamistus remains in the pupal stage for 10 to 12 days, after which the adult moth emerges and are ready to mate. Pupation mostly takes place within the stem, but can also occur between leaves. As mentioned, moths are active during nighttime and live between 2 to 14 days. The life cycle of Sesamia calamistus takes 53 to 54 days to complete when kept in a growth chamber at 28 degrees Celsius and a relative humidity level of 65 to 70%. In general, under actual environmental conditions, the life cycle may take 6 to 10 weeks to complete. Now, this is important. Unlike the previous 2 stalk borer species covered, larvae of Sesamia calamistus do not enter into diapause, but develop throughout the year, even during the dry season. However, under drought or cold temperatures, development slows down and the pest is less abundant, occurring mainly on wild host grasses. Under irrigation, development is uninterrupted. Why? Because this creates a conducive microclimate for continued female moth egg laying and larval development. Because the fall armyworm evolved in a tropical climate, it does not have the ability to diapause and cannot survive in all cropping regions during the winter, such as the high veld, where winter temperatures are too low. However, it should be remembered that moths are migratory and are capable of flying distances of over 100 kilometers in a single night, aided by wind in search of favorable conditions and maize plants. For this reasons, the consequences of the pest differ between geographical regions and seasons. Northern Limpopo, the Lowveld, and northern KwaZulu-Natal are considered hotspot areas, as the relatively high temperatures enable it to overwinter. The regions bordering these hotspot areas are medium risk. In other areas such as the Eastern Cape and Western Cape provinces, sporadic infestations late in the cropping season may be expected. From this, we can already see that the pest status of this species will differ between regions, and this will be discussed in more detail under management strategies. Female moths lay around 100 to 200 eggs on leaves, mostly on the underside, but also on the upper side and on stems. The young instars feed superficially on the outer leaves, usually on the undersides, first feeding at their hatching site and then spreading out throughout the maize plant where they consume the leaf tissue, except the veins and midribs. Newly hatched larvae are difficult to identify morphologically as they closely resemble those of other noctuity moth larvae. The leaf wall is preferred in young plants, whereas on mature plants, they may infest ears where they feed on soft tissue like kernels, soft inner leaves, and silk hairs. They rarely feed on older mature leaves. This is not surprising since this species is described in the scientific literature as a late season pest that shows preference for young late planted maize, particularly in regions that it migrates to. Before I move on to the older larvae, many of you may be wondering as to how the newly hatched larvae spread to other sites. The answer is simple. These small larvae spin silken threads which can then catch the wind to transport larvae to other feeding sites. This behavior is called ballooning and can transport newly hatched larvae over considerable distances. Ballooning is one of the reasons why infestation levels of a field can reach 100% and why larvae may also be found on unrelated crops nearby. With that said, during meetings I attended when I worked at KZN Guard, it was noted that if maize is in close vicinity to other crops, the pest may feed on other hosts, such as potato, but will not attack them per se. In other words, the species will not cause economic damage per se. This has been demonstrated in research conducted by the Agricultural Research Council's Vegetable and Ornamental Plant Institute. In addition, from working closely with extension services in KZN, all reports received pertaining to this pest were to do with maize. Now, the older mature larvae are cannibalistic and often tend to eat each other when in contact at high infestation levels, which reduces the number of mature larvae per plant and in turn reduces the competition for food. The fifth and sixth instar larvae feed in the wall in maize plants. After about 14 to 22 days, the fully grown larvae drops to the ground. As with all lepidopteran maize pests, the exact duration of the larval stage is again largely dependent on temperature, but also diet. And what that means is availability of plants in the right growth stage, in this case, young maize plants. The optimum temperature for larval development has been reported to be 28 degrees Celsius. The fully grown larvae will then burrow 2 to 8 centimeters into the soil before pupating. If the soil is too hard, then the larvae will cover itself in leaf debris before pupating. An adult moth will emerge from the pupae after 7 to 14 days, depending upon soil temperatures in warmer months and around 20 to 30 days in cooler areas. Now, in most parts of South Africa, infestations are usually due to Spodoptera exempta, moths migrating from warmer countries to the north, such as Zambia, Zimbabwe, and Mozambique. Small outbreaks occur frequently in the high rainfall areas of Mpumalanga and KwaZulu-Natal, but a large outbreak occurs basically only once every 6 to 10 years. The worst outbreaks in South Africa occur after late summer rain following drought. Their main migration flight starts in the evening and the insects ascend to 300 meters to 1,000 meters above ground level, moving downwind on the prevailing air currents. Now, as I mentioned earlier, what many notice with regard to this species are large areas of lawn, pasture, or grass crops such as maize and sorghum, covered in dense colonies of larvae, which can virtually happen overnight. So why is this the case? African armyworm outbreaks are known for their unexpected sudden appearance, and this has led to the common name mystery worm. The name armyworm describes the way the larvae crowd together, often marching in the same direction in search of food, hence the name armyworm. In Afrikaans, the African armyworm is called commando worm or commando worm because the large numbers of larvae advancing basically look like a group of commando soldiers on a maneuver. An outbreak is more likely to occur if crops have been sprayed with high quantities of nitrogen, as this causes green, sappy growth, which is very attractive to African armyworm larvae. There were 2 areas in KZN which I was called out to during March 2020, where I witnessed this armyworm behavior that I just described. The outbreaks followed rains late in the summer. The areas were in Msinga and Mfolozi. The larvae were found on grass that was being used to graze rural livestock. The rural communities were quite surprised and in panic, given that this was the first time they witnessed this type of armyworm outbreak in their area. And with that, for all extension officers and crop advisors, it is important that when farmers phone you in a panic, that you calm the farmer down, get to the site, assess the situation, and brief the relevant individuals on the way forward. Do not create more panic. If you do not have the answers, then consult or query with someone that does. Stay calm and sort it out. Finish and clear. So now all of you are probably wondering what happened after I got to the site and how was the situation dealt with. Stay dialed in. The beans will be spilled under management strategies. Now, the female moth lays her eggs in batches of between 100 and 400 and can produce up to 1,000 eggs in her lifetime. These hatch within 3 to 6 days, depending on air temperature and humidity. The larval stage lasts 14 to 32 days. Larvae require temperatures of between 24 to 32 degrees Celsius to develop, and therefore anything below this will hinder development and often cause death of the larvae. Fully grown larvae bury themselves in the ground to pupate. After about 7 to 21 days, the moths emerge and will probably move off and not produce a second generation in the same place. Finally, like the fall armyworm, the African armyworm cannot diapause and will therefore continue to migrate to favorable environments. Right, so with that, we have now covered the respective life histories. Now, as I have been talking, I have mentioned the word feeding quite extensively with regard to the larval stages for the stalk borer and armyworm species we are covering today. It is important that everyone here today understands that it is the larval stage which damages crops through feeding, particularly during the early stages of crop development. This is why the larvae are commonly referred to as the pest destructive stage of a lepidopteran pest life cycle. In addition, this is why management measures target the larvae either directly or indirectly. Registered insecticides, for example, are aimed at controlling the larvae. Spraying the moths is a futile exercise since they are mobile and pesticides generally will not penetrate their fine body scales. Now let's have a look at what feeding damage for each of these moth species looks like. But before that, please give me a sec just to have a quick sip of water. Okay, so let's start with the African maize stalk borer. First instar larvae feed in the young terminal leaf walls, resulting in characteristic patterns of small shot holes as pointed out on your screens that are neatly arranged across the width of leaves. Second instar larvae then feed into the growing points, which may result in plant death and which shows characteristic symptoms of dry withered leaves, also called dead hearts, as we see here with these 2 images. Extensive tunneling into the stems from the 3rd instar onwards results in long fast-filled galleries, as can be seen here, which weaken the stems, resulting in breakages. For those that do not know what is meant by frass, this is simply a nice way to say insect poo. Now, larvae which bore into the maize cob, as we see here, will feed on the developing kernels, leaving cobs filled with frass, thereby negatively affecting production, which results in losses. The newly hatched larvae of the spotted stalk borer produce characteristic leaf windowing while feeding. I think that's quite clearly shown here in this image. They initially feed in the leaf wall. Early larval stages mine in leaves, causing yellow streaks. And unfortunately, I do apologize about the quality, but that's the best image I could find before they bore down inside the funnel. Young leaves also display a series of pinholes. I think this picture here shows it quite nicely. And like Bassiola fusca, feeding in the leaf wall growing point can result in dead hearts. And this is quite a nice picture here showing that. All the larvae then tunnel into the stems, as we see here, eating out extensive frass-filled galleries, which again, like in the case of Bassiola fusca, can result in breakages. Further, larvae can bore into the maize cobs, and I think that's again quite clearly shown in this image, and tunnel extensively, resulting in production losses. Now, to many of you, it should be easy to see that the symptoms here are similar to that of Bassiola. Now, what about Sesamia? Okay, so the most serious damage occurs at the early plant stages. Feeding larvae reduces yield by killing the growing point, resulting in dead hearts, again shown in this image here, causing early leaf senescence and reduced translocation and lodging due to weakened stems. Larval feeding may cause the entire plant to die back. Sesamia calamistis infestation increases the incidence and severity of stalk rot as well. Larvae do not leave any feeding lesions on leaves, but holes can be seen where penetration took place in the stems. And I think that was shown in the previous slides quite clearly. Larvae tunnel into stems, which results in holes, broken stems, eventual death of the growing point of maize. Frass is also deposited in the stems, and that's pointed out here in this image, where larvae feed on kernels. In the cobs, frass deposits are also visible, and I think that's quite clearly shown in these two images here. Now, from this, it should be clear to all of you that the symptoms here are similar to both Bassiola and Kylo, where larvae tunnel into stems and cobs are targeted. Okay, moving on to the fall armyworm. Feeding by the young larval instars 1 to 3 results in semi-transparent patches on the leaves, also called windows, and that's pointed out on all of your screens. Once the 4 to 6 larval instars reach the protective region of the wall, this is where most damage occurs, which can be seen by the presence of ragged holes in the leaves. Large quantities of frass larvae poo is also present, and that can also be seen here in this image. When the frass dries, it resembles sawdust, as most of you would have gathered by now. Feeding on young plants can kill the growing point, resulting in no new leaves or cobs developing. If the plant is older and has already developed cobs, then the larvae will eat its way through the protective leaf bracts into the side of the cob, where it begins to feed on the developing kernels. And I think this image points that out quite clearly. And yeah, again, as all of you can see, leaving behind frass, which ultimately can result in production losses. Okay, so Spodoptera exempta larvae, again, clearly pointed out in these images in the gregarious phase, are known to migrate into maize fields from surrounding vegetation once that food source is depleted. Larvae can consume whole leaves, leaving only leaf veins in some instances. If ears are present in plants, the silks and tips of ears can also be damaged. Solitary African armyworm larvae often feed on maize seedlings and grasses, leaving holes. For me personally, when I witnessed the outbreaks on grass in Msinga and Mfolozi firsthand, I could not visually conclude a significant difference Of course, one has to remember that when the larvae are fully grown, most of the damage would have already been done by the younger instars. However, my initial observation was confirmed by the district scientists who also did not observe a significant difference from the time of the outbreak to the point where larvae were burying themselves in the soil to pupate. Damage in the form of holes were observed on maize plants in direct path of the marching armyworms, and unfortunately I cannot share those images as I no longer have access to them. Now, although fully grown larvae were on the plants, no additional damage could be observed. Overall, while I have witnessed outbreaks, I have not witnessed significant damage to crops, pastures, or grasses. And remember, this is based on my experiences. Others may have had different experiences. I think I may have given away a big clue as to how the situation was dealt with. As noted earlier, I mentioned that under management strategies, I will touch on what was done in terms of control. Before we go any further, let us take a step back and discuss what we currently know. With that said, we need to be practical in how we look at differences between species in terms of the bigger picture and how this knowledge will make a difference to those working on the ground. Now, from covering the larvae, we know that morphologically the mature larvae can be distinguished. We also know that while Bassiola and Chylodiopause during unfavorable conditions, Sesamia calamistis cannot and persists throughout the dry seasons, albeit developing a lot more slowly during drought and cold weather conditions, mainly on wild host grasses. In terms of damage, the neatly arranged shot holes across the leaves is clear evidence of the presence of the African maize stalk borer, whereas yellow streaks in leaves displaying pinholes is evidence of the presence of the spotted stalk borer. Further, Sesamia larvae do not leave behind any feeding lesions on leaves and is considered important, particularly around coastal areas. Overall, Sesamia is not considered as economically significant when compared to Bassiola and Chylo in the country. Although closely related, fall armyworm and the African armyworm have different behaviors and ecologies. Fall armyworm rarely displays the armyworm behavior of larvae massing and marching across fields. Personally, I have not witnessed that, and I don't know of anyone who has in South Africa. In terms of the morphological appearance, while the inverted Y is a feature for both species, the 4 black spots forming a square on late instar fall armyworm larvae is unique. When it comes to feeding damage, the window symptoms and ragged holes on maize leaves are characteristic of fall armyworm in the country, whereas outbreaks of the African armyworm tends to be identified on grasses and pastures, rarely on crops, and not forgetting the easily visible gregarious crowded black larvae. African armyworm, because it is indigenous to the continent, faces many natural biological enemies, including predators, parasitoids. And what do we mean by parasitoids? Well, these are insects whose larvae develop within the bodies of arthropods, eventually killing them. But not only parasitoids, but also diseases. In contrast, fall armyworm is likely to have arrived in Africa unaccompanied by its natural enemies, which enable populations to increase unchecked. However, researchers have found one local parasitoid that attacks the larvae. But caution must be exercised here that attacks the eggs. But yeah, caution must be exercised here in that this egg parasitoid by itself is not enough to control fall armyworm. Finally, let's consider stalk borers versus armyworms. As covered earlier and as touched on, now the larvae, while difficult to distinguish between newly hatched early instar larvae, the later, more mature larvae can be distinguished between all species covered today. In particular, stalk borers lack the 4 black spots forming a square, as is the case in the fall armyworm. And although stalk borers have an inverted Y shape, as can be seen here and also pointed out on their heads as well, it is a lot darker in color compared to that seen on the larvae of the fall and African armyworms. In other words, it is not easily visible to the naked eye. The damage resulting through larval feeding can also be distinguished between species. For example, shot holes versus window symptoms and ragged holes. It is also important to note that the armyworm species covered are migratory, and although the pink stalk borer does not diapause like Bassiola and Kylo, it does not migrate, but rather development slows. Further, the larvae of stalk borer pupate in the stems of maize plants, whereas the African and fall armyworm fully grown larvae drop to the ground and pupate in the soil. Again, let's not forget the armyworm behavior displayed by the African armyworm compared to stalk borers. Okay, so for all of you here, let me quickly tell you about my experiences at the KwaZulu-Natal Agricultural Department to put into context the importance of detection and monitoring. So whenever extension advisory officials or farmers came to me to report a disaster, as they called it, due to a pest, whether insect or disease that targeted a particular crop, it became clear to me that all situations could have been avoided where detection monitoring protocols in the form of pheromone traps, for example, or physical scouting were in place. In all such cases, I dealt with the first question I asked was whether the crop was being inspected for pests or whether there were other detection protocols in place to identify a potential pest problem early. The answer was a resounding no. And to me, this is where a lot of producers fall short, particularly your smallholder producers. More often than not, the producers had to start again or had to rely on much higher usage of pesticides to get their infestation outbreak under control. The other question I commonly asked was whether the crop was inspected to determine whether the initial application had been successful, or in situations where pheromone traps were employed, whether records had been kept on moth counts. Again, the answer I got was a flat no. In pest management, the importance of detection monitoring cannot be overstated, not only to detect a pest problem, but to assess the effectiveness of management strategies. This forms the basis of any integrated pest management program. Now, when it comes to extension and advisory officials or crop advisors, agents, these individuals need to be a jack of all trades. In other words, they not only need to have an adequate understanding of insect pests, but also diseases and weeds, as well as understanding the biology of the crops they are advising on. For this reason, they require information and knowledge that is beneficial for on-ground or farm situation. Let's take the eggs for the pests we have covered today. Many information sources online promote the scouting of eggs. My question here is, of what value is this to the farmer or his scout? How would they realistically differentiate between eggs? And remember, this is not an easy task for entomologists as well. If the identity of the pest problem cannot be established, how does one treat it? So for me, scouting for eggs is not ideal or practical. Just ask yourselves honestly, what would the farmer's reaction be, and would this be done? I guarantee you that you will be told that you are wasting your time in many instances and forget any action in this regard. This is something that extension advisory officials noted to me during a training session when I was with KZN DARD. Before I even started, they pleaded for knowledge that will help them fulfill their tasks in providing accurate advice and assistance. What they did not want was for me to talk about morphological differences between each larval instar for each pest, because that becomes academic and is important from an entomological research perspective to establish such information. They wanted the key distinguishing characteristics because they understand that a farmer cannot be drilled with technical information but needs to know what matters. Scouting for pupae also makes no sense given the practicality of this and the difficulties in accurately distinguishing between species and the fact that this stage also follows the larval stage. As all of you now know, it is the larvae that are the pest destructive stage when it comes to lepidopteran pests of maize. Therefore, scouting for larvae is practical, but not just the larvae. While it is of vital importance to get the identity of the pest right in order to determine suitable corrective actions for the crop pest situation, what most farmers will see first is the characteristic damage symptoms, and that will give them a clear indication of the culprit. It is important to identify any pest infestation as early as possible because, as touched on, this will have control implications. Let's have a closer look at how producers can scout for stalk borers and the armyworm species that are subject of today's webinar. Every field entomologist will tell all of you that scouting constitutes one of the best weapons producers have in their arsenal to combat the threat posed by stalk borers and fall armyworm. In particular, scouting really means walking through your lands and inspecting whichever crop is relevant within an integrated pest management system from pre-planting to harvesting and beyond for some important aspects of whole production management cycle. Formally, scouting is defined as rapidly and systematically determining overall crop health and estimating presence of certain organisms causing damage and potentially yield reduction. The word identification is key to me. It is important to get the identity of the pest problem right the first time. This will ensure that the appropriate corrective interventions are implemented only when required. For this reason, it is vital that farmers understand the concept of the economic threshold level to determine spray interventions in this regard. The economic threshold level is defined as the population level where control must be applied to prevent the insect population from reaching the economic damage level. However, farmers must take note that control is not an option when the cost of application exceeds the loss in value of the crop caused by a pest. Regular monitoring enables growers or their scouts to gather accurate, up-to-date information on the identity and location of pest problems, as well as making a sound assessment as to the extent of infestation damage based on threshold levels relative to the scouting techniques being employed on a particular farm. No two farms will be exactly the same and experience the same problems. It is not a one-shoe-size-fits-all. What this means at the end of the day is that farmers will have to understand the population dynamics of key pest species on their individual farms. This can be done in consultation with suitably certified pest management personnel for accurate interpretations of population pressure based on monitoring information. Again, to reiterate, this is the most essential part of an integrated pest management, commonly known as IPM program. The best method to identify whether there is a stalk borer infestation in your maize is to scout and inspect your land. Farmers can scout in a grid pattern to get an overall picture of the field. Once scouts have finished scouting the field, farmers should try and quantify the damage into a percentage, which will help them determine if it would make economic sense to spray an insecticide. How do you look for stalk borer infestations? The first signs of early damage usually noticed are the small holes or shot phase caused by the smaller larvae on the leaves of maize. These holes resemble the effect that would be caused by the spray of shotgun pellets if shot at an object, the characteristic symptoms associated with Bassiola fusca, as you all now know. In the case of Kylo, remember that what I said when we covered damage, early larval stages, mine in leaves causing yellow streaks where leaves also display pinholes. Now, if these are observed for either species, you probably already have an infection rate that will exceed 10% damage to the crop, and therefore corrective action will be required. The 10% damage threshold is a general rule of thumb used by farmers to indicate to them when corrective action becomes necessary. This will become clearer when we look at insecticide label examples under management strategies. Now, as their name, stalk borer, suggests, the larvae feed primarily in the stalk of the maize plant. Dead hearts is also one of the latest symptoms to be noticed by farmers, as the central leaves at the top of the plant become drier and withered and may even flop over to the underside of the growing maize plant. You have all seen images of dead heart symptoms earlier. While this is characteristic for all stalk borer species, remember that Sesamia, unlike Bassiola and Kylo, leaves no feeding lesions on leaves but holes in the stem, indicating where penetration took place, which are telltale signs of the presence of Sesamia, particularly for farmers in the coastal areas, given my comments earlier. At the tasseling stage, the larvae do also sometimes drop onto the maize cob and feed on the cob itself or at the base of the ear. If this is the case, a farmer can sometimes identify premature fallen ears of maize, cutting off the energy supply to the ear of the maize. Another method of identifying infestations is for scouts to look for the physical larvae in the plants, but as mentioned earlier, this must be linked to early characteristic damage symptoms as well. Where the identity of the pest problem cannot be established, farmers must seek advice from their respective advisors. Let's move on to the fall armyworm. One of the most important things that farmers can do to manage fall armyworm is to enter their fields at least once a week, more often when there are dynamic changes. For fall armyworm, here is an example of a scouting protocol developed by the Food and Agriculture Organization, better known as the FAO, and supported by DLAD. That is quite simple. What this protocol says is that a farmer must firstly determine the maize field to be sampled. If the fields were planted at different times with different varieties or with different conditions, then each plot should be sampled differently. So in the field, and as pointed out on your screens, walk a letter W covering the entire field, as all of you can see on your screens. At the start, at every turn, and at the end, inspect 10 plants in a row. These 10 plants are called a station. Look carefully in the wall of each plant for signs of the characteristic window symptoms of fresh frass. These indicate a live larva, probably fall armyworm, in the wall. Do not include plants with some damage to older leaves, but with no clear signs of recent damage. Only recently infested plants need to be counted. Keep track of the number of plants currently infested in this way. This method may also be used by farmers to scout for the characteristic early feeding damage associated with stalk borers as well. Now let's take a hypothetical example. The total number of plants infested by fall armyworm in the 50 plants counted is 26. So in 100 plants, it would be double. 26 multiplied by 2 would equals 52 or 52% of the plants infested. Because we are looking for early signs of fall armyworm presence, in other words, the window symptoms of frass in the wall, the sampling doesn't depend on finding the larvae. Remember what I said about the newly hatched larvae? Instar, they are very difficult to distinguish between pest species in the context of maize. Another advantage is that the sampling is fast, non-destructive, and can be done at any time of the day. Guidelines to economic thresholds for the fall armyworm indicate that control should begin when 5 to 10% of young maize plants in the early stages of development show signs of damage symptoms, 20% of plants in the mid-wall stage, and 40% of plants in the late vegetative growth stage. The latter applies to maize plant seedlings that will not be subjected to reinfestation. For the reproductive stage, a threshold of 20% may be considered. So why a threshold of 5 to 10% early and 20% during the reproductive stages of maize development? Firstly, research has shown that maize plants infested by fall armyworm during the very early stages, 1 to 3 weeks after seedling emergence, suffer high yield loss. In addition, we know that the protective leaf bracts around the developing cobs are attractive to larvae, where this can result in direct injury to the developing kernels, leading to production losses. Now, while I have noted these guidelines, remember my comment earlier that it is not a one-shoe-size-fits-all. Let's go back to the 52% estimate of plants infested, and let's say the maize crop was in the mid-wall stage. Based on the threshold of 20%, the corrective action on this maize field would need to be immediately undertaken. Moving on to the African armyworm. Now, my question here really is, given what we have already discussed around the species, can one really effectively scout for this pest given their sudden unexpected appearance and not forgetting their armyworm behavior? For me personally, I do not think so. In general, large numbers of birds congregating on the veld is usually a good indication of the presence of African armyworm. Okay, So now this is where it gets interesting for most who have dialed in, because this is now really where all the pieces of the puzzle come together under management strategies. So before we get stuck into it, if you just let me have a quick sip of water. Okay, thank you. Let's get into it. So with Stalk Borers, given their life histories, management options are pretty much the same. First and foremost, I want to say to all of you, growing healthy crops with balanced fertilization that receives adequate moisture is paramount so that crops can better withstand pest damage. Now, very quickly, for those here today who may not know what cultural methods of control are, simply put, cultural methods are agricultural practices used to enhance crop health and prevent weed, pest, or disease problems without the use of chemical substances. Manipulating planting dates by smallholder farmers in particular can be implemented to avoid periods of peak moth activity. However, for those planting under dryland conditions, planting dates are usually determined upon the onset of the first effective rains, and therefore this may not always be an option for every farming system. In addition, a fair knowledge of the population dynamics of stalk borer species present on individual farms is required to get the timing right for this to be effective, in my view, as well. Good crop hygiene involves destroying maize residues by burning to eliminate larvae and pupae within the stems, and removal of volunteer crop plants and/or alternative hosts. This prevents carryover populations, which helps in limiting the initial establishment of Bacilliola and Kylo that would infest the next crop. Again, from a no-till perspective, this will not be considered since the crop residues are required to protect the soil from erosion. But with that said, let's not forget that larvae of Bacilliola and Kylo can diapause in such debris, thereby carrying over larval populations from one season to the next, which has been recorded. This simply means that no-till farmers can expect higher infestation levels where these two stalk borer species are present, leading to possibly increased insecticide use. Planting early maize in large blocks spread around the field will entice female moths to lay their eggs before the main crop is up, or as they say. This trap crop has to be used for silage before the second generation develops. Now, for me, this is not a practical option for any level of farming. Why? Because to me, it does not guarantee that the main crop will not be infested, and this could be due to a number of reasons. It could also present a waste of financial resources that would be needed to get this operation done. To my knowledge and from my experiences in KZNs, no farmers I spoke to ever considered this option. In general, it has been reported that maize sown as a trap crop was not effective because late rains delayed germination. Further, there does not appear to have been any further serious consideration of this technique after the 1930s. Moving on to the push-pull strategy. So the push-pull strategy involves using repellent push plants and trap pull plants. Let's look at a practical example of a push-pull strategy that I briefly touched on during the early parts of this webinar. In this example, maize is the primary crop. Desmodium is planted between rows of maize. Grasses such as vetiver, napier are planted around the perimeter of the crop. Volatile chemicals associated with desmodium repel stalk borer moths. The repelled stalk borer moths then become attracted to the grass surrounding the maize field, where their eggs are laid. Since the emerging larvae are unable to utilize vetiver, napier grass, they starve to death. This reduces damaging stalk borer populations. Now, from a practicality perspective for farmers, this is not practical, simple as that. While it may sound nice on paper, it is a different story in the field. Does it also guarantee that insecticides will not be required? I don't think so. Commercial farmers will not entertain this option, and neither will smallholder farmers either. Again, I have spoken to quite a few maize farmers in the KZN province, whether small or large scale, and one of their comments were as follows: "You must be dreaming," but when this topic came up. Certainly, if I were a farmer, I would be thinking the exact same thing. Management has to be practical but sound at the same time. Now, tillage in the form of deep plowing to bury maize stubble was one of the earliest control measures used against this pest in South Africa. Why? Because the larvae of Bassiola and Kylo are in a state of suspended development and therefore present the weak link in the life cycle. This action reduces subsequent threats. Again, in a no-till situation, this will likely not be done. Hence, no-till farmers. Hence, for no-till farmers, the incidence of the African maize stalk borer in particular is increased. The only practical method to monitor for moths is with the use of pheromone-baited traps. And here's an example of a pheromone bucket trap. The key benefit for farmers being that pheromone traps detect the pest on arrival in a particular area and can be used to gather information pertaining to the population dynamics of the target species relatively easily. Currently in South Africa, there are no pheromone lures registered to monitor for stalk borer species. To monitor for stalk borer moths, and therefore relying solely on physical scouting, as we've discussed now, will be to identifying an infestation early for corrective action to be effective. Finally, let's have a look at insecticides. Now, you have heard me use the words corrective action, but I've not elaborated on this further. In every case that I have dealt with, corrective action involves spraying the target pest with the pesticide. Again, to reiterate, scouting has to be on point to identify stalk borer infestations early and determine if spraying will be feasible based on threshold levels. A suitable registered insecticide should be applied when characteristic feeding marks are evident in the youngest leaves and larvae have not yet migrated into the stalks. As mentioned, if 10% or more plants exhibit the characteristic leaf damage symptoms, then the primary method of control is by insecticide application. A second successive insecticide application may need to be applied 2 weeks after the first application because larvae that hatch more than a week later may survive on the maize plant because of a sublethal residual concentration of the insecticide that remains. This is particularly important in the case of Bassiola. The successive application 2 weeks later will ensure that any surviving larvae are also killed. Numerous insecticides are registered for early corrective post-emergence treatment. If late corrective treatment is required, and this is generally when the larvae have started to tunnel into the stems, a suitably registered insecticide will have to be used. So what do we mean by systemic insecticides? Well, these are insecticides which are taken up by plants and ingested by pests that feed on the plant material. After ingestion of plant material, these chemicals then serve as stomach insecticides. Pre-emergence preventative treatments involve applying a registered insecticide to the soil together with the seed. Now, treatments in this regard are expensive and should only really be considered if a moderate to high yield of maize is expected. Now, for me, the question that I was asked most often during my days at DARD by farmers was, what chemical do I use? Where do I find this information? Well, for all of you here today, my answer should not be a surprise. The best platform to find accurate, up-to-date information pertaining to registered pesticides in terms of crop and use is AgriIntel for me. AgriIntel was my go-to option on my cell phone and computer, where I created a shortcut to the website. And without it, I can tell all of you now, I would not have been able to advise farmers accordingly. AgriIntel really made my job a lot easier. Okay, so here we see an example of an insecticide label registered for use against both Bassiola and Kylo. There are different instructions in terms of dosage rates when it comes to ground applications, where there is a difference of 0.1 mol per 100 meter row between the two species, so they are not exactly the same. Note that this product can also be applied aerially for Bassiola fusca. Now, what's really key here are the remarks for me. Okay, so as you as you all will see, thresholds for spraying this product are clearly stipulated for both pests, for both species. And in addition, the remarks clearly state that the spray must be directed into the maize funnel as well as adjoining leaves in the case of Kylo. Also take note of the statement that mature larvae larger than 1 centimeter that have already bored into the stalk will not be satisfactorily controlled. Therefore, for this product to be effective, an infestation must be identified early before the larvae tunnel into the stalks. Pay careful attention to the reapplication notes as well. Be sure to scout recent damage on the newest 2 to 3 leaves for this purpose. Okay, here we see a label example for a product registered specifically for the pink stalk borer. We also see that this product can be applied aerially for this purpose. The remarks are also clear in how the product must be utilized to be effective. Now, this label is similar to the first one I showed all of you. However, the dosage rates to be applied are the same. The damage threshold for spraying is also similar, but we see that in the case of Barceola, a second application may be required 10 to 14 days later. Because like I said earlier, larvae that hatch more than a week later may survive on the maize plant because of a sublethal residual concentration of the insecticide that remains. Again, the label makes it clear that the spray must be directed to the funnels. Here we see an example of a label that is registered against stalk borers on maize, and unlike previous labels, no species are noted. There we go. Therefore, from an interpretation perspective, this product can be used on stalk borers regardless of the species present on the maize crop. Again, this product can be used aerially as well. Interesting, unlike the previous product labels, instructions are based on plants infested with eggs. From a practical standpoint, farmers or their scouts, as mentioned earlier, are not going to scout for eggs in most instances on maize plants. I do not know personally of any who do. So could one then work on a damage threshold of 10% as we have seen on the previous label examples where a 5% egg infestation threshold was also noted? I'm not sure, but it would make sense. This is something that can be queried with the individuals selling the product. Okay, so lastly, this label is similar to the previous in that no species are noted. However, it is different in that this product can be used preventatively at planting time of maize and not just for corrective purposes for farms where stalk borers present a legitimate threat. Also take note of the reduced tillage remarks, where it is noted that pest populations in this instance may be higher, as I touched on in the previous slide, and therefore it becomes important to monitor the degree of control of the first application, and if necessary, the product will have to be reapplied. It is very important to also take note that follow-up rains or irrigation is essential for adequate absorption and translocation. In other words, if there is no rain or irrigation after application, the product will likely lack the required efficacy needed to control stalk borers. Again, to reiterate, this type of preventative option should only be considered if a moderate to high yield of maize is expected, given the costs involved. Now, to me, it is important that anyone in an advisory role provides options in line with the situation the end user is faced with. The end user being advised then has the opportunity to weigh up the options and determine which best suits their situation, available resources, and wallet. I go back to a previous comment. It is not a one shoe size fits all. So what about genetically modified maize? And for all of you who have been listening carefully, we'll be asking that exact question. Over the past 20 years, South African maize growers have rapidly embraced biotech crops with built-in insecticidal proteins that provides protection against stalk borers. Biotech maize varieties expressing a range of different insecticidal proteins from the naturally occurring soil bacterium, Bacillus thuringiensis, have been commercially approved by South African regulators as single or stacked Bt traits. The proteins taken from the bacteria Bacillus thuringiensis are toxic to stalk borers. With that said, it is worthwhile keeping the following in mind. BT maize seed costs about double the price of standard non-genetically modified seed. However, the effect it has on lepidopteran pests can be of great economic benefit to farmers as well. It is crucial that the area planted with BT maize includes the planting of a mandated refuge to prevent the buildup of insect resistance to the toxin. It is vital to farm responsibly and include crop rotation to allow a change from using BT maize and broadleaf crops on a continuous cycle. These factors and the fact that BT maize varieties are often not suited to planting in suboptimal agricultural environments prevent smallholders from fully benefiting from this option. But with that said, what about social acceptance of the technology and skill set among smallholder farmers? One could then follow up any answers there with the question of awareness. How many smallholder farmers understand this technology and how to use it properly? From a large-scale maize farming perspective, BT maize is the go-to option for many farmers. Now, as with everything in life, with great power comes great responsibility as well. I mentioned the reason for the planting of a mandated refuge. Before we take a closer look at how to plant a refuge area, let me quickly touch on how resistance evolves and how resistance can be managed in this context. A small number of insects in the population are naturally resistant to the BT trait. When maize with the BT trait is initially used, it controls almost all of the insects in the populations. Survivors are resistant to the BT trait and lead to the next generation of resistant insects when they mate with other resistant insects. Similar to other crop protection products, insects can also adapt and develop resistance to biotech crops with inherent BT insect-resistant traits. The main factors contributing to these cases of targeted pest resistance has been the cultivation of BT crops for pest management without the implementation of proper resistant management practices, including not adhering to the requirement for the planting of a crop refuge. Key components of resistance management for BT maize varieties include scouting, crop rotation, stacked traits with multiple modes of action, and again, the mandatory planting of a crop refuge. In terms of scouting, farmers must monitor their fields to know what pests are present and assess crop damage to help inform their decisions on pest management. When it comes to crop rotation, growers should refrain from planting one type of crop in the same field for consecutive years and rather grow a different crop entirely in alternating years. Alternatively, growers should avoid planting BT crops in the same field 2 years in a row and rather use non-BT varieties. This gives insects less exposure to BT proteins, thereby slowing resistance developing. In terms of stacked traits with multiple modes of action, insects are less likely to develop resistance to BT crops with multiple modes of action compared to a single mode of action because it is much harder to develop resistance to 2 or more BT traits at the same time. Insect-resistant technology with the cultivation of BT crops, as mentioned, is widely used in commercial maize production in South Africa and requires that farmers plant a structured refuge as part of their IPM program. Now, while I have been mentioning a refuge area, I have not quite explained exactly what this entails. A refuge is the mandatory planting of an area of the same crop without the BT trait adjacent to the BT crop field. The refuge then maintains a population of susceptible insect pests not exposed to selection pressure from the BT protein. These susceptible insects would be available in high enough numbers to breed with resistance insects that may emerge in the BT crop field, ensuring that susceptibility is passed on to the offspring and thereby helping to prevent the emergence of resistant populations over time. Now, how should a refuge be planted? Refuge requirements vary depending on the country, crop, and types of pests to be controlled. The refuge management options available to farmers of BT insect resistant crops in South Africa is to plant either a 5% non-BT refuge of the same crop that is not treated with an insecticide, or 20% non-BT refuge that may be treated with a registered non-BT containing insecticide biopesticide when 10% of the plants exhibit stalk borer feeding damage. In addition, growers must ensure that refuge is planted within 400 meters from the BT crop within 7 days of planting of the BT crops, with hybrids having the same maturity of the BT crops and be managed in the same way with the minimum of 6 rows and without any mixing of BT and non-BT seed. Okay, so now when it comes to the fall armyworm in terms of crop management, avoid planting late in the season where possible. This is because the species is more threatening to young, late planted maize plants, as noted in the scientific literature. Moreover, avoid staggered planting where possible, as well, as this will continue to provide the favored food for fall armyworm locally, the young maize plants. This will ensure that plants have enough vigor and will be able to better withstand infestation and feeding damage by fall armyworm larvae. Currently, these are the most important recommendations for smallholder farmers with regard to this pest. Ensuring good soil health and adequate moisture to grow healthy crops that can better withstand pest attacks cannot be overstated. Another detection method is the use of pheromone-baited traps to detect and monitor for armyworm male moths early in growing seasons. Unlike stalk borers, there are 2 choices of commercially available pheromone lures that can be placed into bucket traps and used to monitor the activity of male moths on farms. Both lures, one a rubber septum and the other a bubble cap, contain the active ingredients Z7 dodecyl acetate, Z11 hexadecyl acetate, and Z9 tetradecyl acetate, which simply put is the female fall armyworm sex pheromone, which attracts male moths to the traps. As the moths fly down the funnel and into the bucket in search of the female, they come into contact with Vapona dichlorphos DDV-based strips that have been placed into the bucket, which kill the male moths and allows them to collect in the bucket. So why am I referring to DDVP strips instead of what has been done generally in the past using dishwashing liquid and water? Why? Because with this method here, you get a better chance to get the morphological identification correct. So based on morphology, you want to preserve that. Okay, so trap catches should be recorded weekly, preferably on the same day every week. The trapped moths must be removed from the traps after counting, and these catches of fall armyworm male moths are an indicator of infestation and should be used to determine threshold for insecticide spraying programs. It is essential, in my opinion, to follow pheromone label instructions for this purpose. With that being said, pheromone trapping is not a substitute for scouting, where the two should be combined for best results. Before we move on to insecticides, and given what I said when I covered life history, the pest status of fall armyworm differs between areas where it is permanently established and areas where it migrates annually. Maize planted in areas where the pest is permanently established is always at risk of infestation, as high-risk areas expect continual infestations to occur. It is also important to start and stop applying the chemical precisely according to the larvae's developmental stage. Insecticides generally will not be effective against older larvae larger than 1 centimeter. Fall armyworm control programs in medium and low-risk areas should be guided by the principles of integrated pest management. As the pest cannot survive in these areas, it migrates from the overwintering areas to the medium and low-risk areas during spring and summer. It is impossible to determine when or even if a serious infestation will occur. Preventative control is therefore not an option. It is no use applying an insecticide against an insect that has not yet infested the crop. That just represents a waste of time and resources to me. The decision on when to spray should be based on regular scouting results and thresholds set for a farm. There are various insecticides registered to control fall armyworm larvae. Given the importance of resistance management for this pest, insecticides from different mode of action groups will have to be rotated, particularly in high-risk regions where intense spraying programs are likely to be the order of the day. In this regard, it is very important to follow the label instructions with regard to resistance management when it comes to the fall armyworm. We will look at label examples in the next slide, but before that, what about BT maize? Is it effective against fall armyworm larvae? The short answer is yes. The Agricultural Research Council, in collaboration with Northwest University, conducted trials to determine larval susceptibility to BT maize. Larvae were placed into maize walls of non-BT maize plants, the control groups, as well as into first-generation BT maize commercialized in South Africa, BT1, and second-generation BT maize, BT2. 95% of the larvae on the control maize plant survived after feeding for 14 days, 47% on BT1, and absolutely null on BT2. The research concluded that BT1 will suppress fall armyworm, while BT2 has the potential to control larval infestations. But there is one big problem. There is no formal registration, and therefore the technology cannot at this point in time be considered in the management of fall armyworm. It's unfortunate, but yeah, that is the case. Okay, now from this first label example, we see it is similar to the last label I showed all of you for Stalk Borer. How so? Well, for this product, the label notes Spodoptera, as we see here, Spodoptera species. The SPP simply stands for species. And therefore, this product can be used against any pest species that belongs to the genus Spodoptera, which targets maize. This product can be applied both aerially, by ground, according to the label dosage instructions. Under the remarks, we see that they note the early feeding damage signs, the small windows in the leaves. I don't think that's quite enough to make a decision on spraying, but with that said, all of you should be well aware that the decision to spray should be based on monitoring scouting records. Now, let's take a look at a hypothetical example here. A farm where a 5% threshold is set based on the early damage symptoms of fall armyworm on young plants will need to spray as soon as this threshold is breached based on scouting records where small windows in the maize leaves are observed. Remember what I said earlier. Insecticides used against fall armyworm is not an option when the cost of application exceeds the loss in value of the crop caused by the pest. Further, based on what I have just covered on the previous slide, the threshold may be set at 10% for areas that are not at high risk for the same stage of crop development. We also see that at the reproductive stage of crop growth, the product must be applied while the larvae are still outside the cob, because once they move in, the product will not be effective. Now, very important, right at the bottom, as we see here, what we see, and I've just given each of you a second to pay attention to that part. What we see is that this product can be applied twice to a block of maize, where each application is spaced 7 days apart. Where more than 2 sprays are required, the label makes it clear that the product must be alternated with another insecticide from another mode of action group, where 2 examples of products are provided as well. As per the resistance management guidelines compiled by the Insecticide Resistance Action Committee of South Africa, better known as IRAC, a farmer must not return to the initial product mode of action group that was used before 60 days. It is no secret that this pest has a documented history of developing resistance against insecticides elsewhere. Farmers need to practice responsible resistance management in order to keep the toolbox full against this pest. With this example, the first thing to me is that it specifies the fall armyworm. Spodoptera frugiperda and therefore, when used according to the stipulated doses instructions, it will be effective in controlling fall armyworm, but is not registered for use against other Spodoptera species. And what this should indicate to a farmer is that this product should not be used for controlling other Spodoptera species because efficacy cannot be guaranteed. Here we see the instructions indicate that the product should be used when larvae are first noted. Again, we know that larvae in the early stages are not easy to distinguish between species, and secondly, we know what the early feeding damage looks like. Therefore, I would stick to scouting for early damage, but I will also look for live larvae during scouting. Where the spraying threshold for a farm has been surpassed, the product must be used. We also see that for follow-up sprays, the decision on whether to spray or not will depend on scouting results. But importantly, any follow-up spray that may be required after the initial spray should be done 10 to 14 days. Now, it also clearly states that 2 applications in a block should not be exceeded. Now, this pertains to resistance management and says to a farmer that after using this product twice in a block, the product must be rotated with another insecticide belonging to a different mode of action group. Unlike the previous label example, it does not highlight any products. In this case, a farmer must refer to the front of the product label and note the IRAC insecticide group code. In this case, it's a 3. Therefore, where a third application may be required before 60 days has passed, another insecticide suitably registered for the crop pest situation with another IRAC insecticide group, for example, 22A, will have to be utilized. This is a very important point to keep in mind. And as discussed previously now as well, a farmer can only return to the initial mode of insecticide group used in a particular block only after 60 days for sprays as per the label. Okay, so one final example, again, very similar to the previous 2, also clearly stated that the product will not successfully control larvae larger than 1 centimeters in length. One thing I want to bring all of your attention to is under remarks. It states that the product should be applied early morning when dew is present. And let me just point that out so that everyone can see that. Here we see it here. Okay. Now, when talking about pesticides in general, it is often considered that spraying before dew has evaporated is bad news, as it will prevent the spray mixture. It will prevent the spray mixture being deposited effectively onto the target and prolong the dry-off period. However, as can be seen with this label example, this is not always the case and will depend on the formulation of the product. The take-home message here is to always apply an insecticide according to the label instructions. With that said, let's have a look at the African armyworm. Okay, so it is important to know that for control measures to be effective against this pest, the worms must be found in time. As I have covered under life history, if the larvae are discovered when they are fully grown, the use of insecticide control is often not recommended, as most of the damage to crops will already have been done, with emerging adults likely migrating to another place to produce a second generation. As I also highlighted, the larvae require temperatures of between 24 to 32 degrees Celsius to develop, and temperatures below this will decrease the rate of development and result in death. Now, where the larvae are moving from one land to another, they can be halted by plowing a furrow with pits dug at intervals. The larvae will crawl along the furrows and eventually fall into the pits where they can be covered up. When it comes to the use of insecticide, spraying is most effective if applied as soon as the worms have emerged and are 1 to 5 millimeters long, as these instars are more susceptible to poisons than older instars. So all of you would be wondering at this point as to how the outbreaks in Msinga and Mfolozi were dealt with. Firstly, let me note key points I have mentioned throughout this webinar. No significant damage to the grass or maize was observed, and more importantly, larvae were larger than 5 millimeters long and were getting ready to pupate. Based on that alone, it should be clear to everyone that spraying was a futile exercise given the situation at hand. Burrows and pits were dug up, and as larvae fell into the pits, they were covered up with soil. In addition, when the two sites were visited a week later, the larvae were almost all gone. Hence, the information that the emerging adult moths will move off and likely not develop a second generation held true. Now, before everyone thinks it is as simple as that, one aspect of African armyworm outbreaks is the poisoning, which sometimes follows an infestation. This has been recorded on kikuyu grass and only affects cattle under field conditions. Symptoms in cattle usually appear about 10 days after the appearance of the worms, and only some after the appearance of the larvae, and only some kikuyu pastures produce this toxicity. Symptoms in cattle are as follows. The swallowing of affected cattle is paralyzed, appearance of large strings of watery saliva drooling from the mouth, and the cattle exhibit an apparent severe thirst. Slight appearances of bloat, grinding of teeth, and nervous twitching may occur. Now, very importantly, as soon as symptoms are observed, all cattle should be removed from the affected pastures and a vet called in. A good prevention of further poisoning is the removal of all animals from the pasture for a period of at least 40 days. Luckily, with the two cases I dealt with, the grass was not kikuyu, given that cattle were roaming freely in the area. Finally, for those farmers who are able to identify an outbreak early where insecticide application will prove effective, are there any registered products? Again, the answer is yes. Let's have a quick look at two product label examples on the next slide. Okay, so now there was only one product I could find when searching on Agri-Intel that was specifically registered for controlling African armyworm Spodoptera exempta outbreaks on pastures. But what about maize? Well, all of you will remember this label example, and given that the African armyworm belongs to the genus Spodoptera, the following product may be applied against this pest on maize, provided the product is identified when insecticide control is still feasible. And remember, these are just certain examples that I've plucked out. There are many such examples of suitably registered products as well. Okay, so when using an insecticide, keep the following in mind. As stated on numerous occasions throughout this webinar, the decision for a second, third, or fourth spray, etc., should be based on diligent scouting of the newest 2 to 3 leaves and compared to thresholds set for that particular farm for that stage of crop development in order to determine whether follow-up sprays may be required. When an insecticide is to be used, try to apply the insecticide early morning or late afternoon. Why? Because this reduces the degrading effects of heat and direct sunlight on the product, but importantly also ensures that the larvae, which are mostly active at night, receive maximum exposure to it. Whenever using a pesticide product, and as highlighted with the examples discussed, the product must be used strictly according to the label dosage and application instructions. Ensure the product is registered for control of the target pest on the crop. When a pesticide is used strictly according to label and provided spraying equipment is in good working order and has been calibrated to apply the correct dosage, water volume, with the correct size nozzle for a particular crop pest situation, users can be guaranteed that the product will be effective in controlling the target pest. It is important to read these instructions carefully. Pesticide companies have done copious amounts of research to compile the label instructions, and if not followed, efficacy cannot be guaranteed. It's as simple as that. While at KZN DARD, regardless of the target pest, many smallholder farmers approached me noting that a particular pesticide product they had purchased was not doing the job and they suspected the product may be ineffective against the pest. However, when technicians were sent out to look at how the farmers were conducting their spray operations, it was usually found that label instructions in terms of dosage and applications were not strictly followed and sprayers were not properly calibrated. In some instances, the product was not registered for the crop pest situation. This puts into perspective that it is easy to forget about the little things, which in the end can make a huge difference. Please keep that in mind, everyone. Finally, you always use personal protective equipment and clothing, commonly referred to as PPE, whenever handling a pesticide product. The risks associated with pesticides can be negated by using the product responsibly. Remember, a hazardous chemical is like a tiger in a cage. Know, understand, and respect the tiger that you are working with so that there are no unexpected surprises. Employers are mandated under the Occupational Health and Safety Act, Act number 85 of 1993, to provide employees handling and spraying pesticide products with the appropriate PPE. Ensure that all employees receiving PPE sign a register which must be safely filed to serve as proof of this. In terms of key differences between stalk borers, fall armyworm, and the African armyworm when it comes to management are firstly, tillage in the form of plowing, which is a good cultural option against the two most important stalk borer species in the country, Bassiola and Kylo. This will not be suitable for the two armyworm species covered today, as these pests will migrate in search of their preferred hosts and favorable environmental conditions for continued egg laying and larval development. When it comes to detection using pheromones traps, this can be done for the fall armyworm given the availability of commercially available pheromone lures. However, for stalk borers in particular, physical scouting will have to be relied upon. Although light traps are effective and can warn farmers of possible Lepidoptera pest infestations, there are some practical limitations in the execution of this method when used for long periods of time. For example, moth numbers caught in light traps may be influenced by climatic events such as the intensity of moonlight, which should be taken into consideration as well. While BT maize varieties is an option for farmers when it comes to stalk borers, this is not an option for fall armyworm, given the fact that there is no formal registration to date. Until the BT maize varieties are officially registered against fall armyworm in South Africa under the relevant acts, agricultural extension officers or any other advisors in the industry are not allowed to recommend the use of BT maize for protection against fall armyworm. When insecticides are used to target young fall armyworm larvae, ensure that resistance management guidelines as per the label of insecticide products used are adhered to. For more information and to stay updated with regard to fall armyworm resistance management guidelines, visit the IRAC website, www.iraconline.org. And finally, from experiences to date, the African armyworm does not seem to pose a major risk to maize in the country. However, passions and grasses tend to be more at risk. Further, cattle farmers using Q need to be vigilant and take action as necessary following an African armyworm outbreak. Okay, so CropLife South Africa encourages all producers to actively scout and keep well-managed pest records in order to maintain crop protection costs at sustainable levels. By keeping records over time of all observations, this will help producers in particular to make informed decisions pertaining to their management strategies, allowing for fine tweaking or adjustments where necessary. Secondly, these records combined with weather data may be useful in predicting infestation patterns in following seasons. Given the current South African economic woes, producers need to be smarter in how they do business, where decisions must be informed by data evidence in order to save on costs in all facets. Now here is an example of fall armyworm and the African maize stalk borer male moth pheromone trap catches on the Sadara Research Station for the 2020-21 and 2021-22 seasons respectively. During the 2020-21 summer growing season, there was a rubber septum pheromone lure available on the market registered for monitoring Bassiola. However, this is no longer the case as already noted. Traps were serviced on mostly a weekly basis and numbers of male moths trapped were recorded. When these numbers are looked at using a simple line graph, the first thing that can be seen for both seasons is that Bassiola clearly is clearly the important pest in the region when compared to fall armyworm, where numbers paled in comparison to that of Bassiola. And I think that is quite clearly evident. This was also confirmed through physical scouting, where most plants exhibited the characteristic shot hole symptoms. It can also be seen that fall armyworm in the region was detected around mid-late December when numbers were fairly low. Spraying against the fall armyworm was rarely done on the station, given that numbers trapped weekly did not meet threshold levels as stipulated on the label of the pheromone brand that was being used. No losses were reported due to fall armyworm, and neither were any cobs damaged during my time on the station. So why were the numbers so low for the fall armyworm in the area? So to me, the first possibility is that the low capture may reflect the low fall armyworm population in the region. The pest status of this species, as outlined in the scientific literature, depends on optimal climatic conditions. Monitoring data from pheromone traps placed on all research stations for the 2020-21 growing seasons, as well as reports from Extension Advisory, indicated that this pest occurs in high numbers in the northern regions of the province, and that's something that's also been mentioned earlier. But for example, areas such as Pongola, Jozini, Makatini, when compared to the central and southern regions of the KZN provinces. Important to note that Sidara falls into the southern region of the province. And these regions are where the species has not been a major concern. This trend was also in agreement with monitoring data collected by Crop Watch Africa. Therefore, climatic conditions appear to be the determining factor. While this constitutes a viable reason, currently there are no published data pertaining to the seasonal abundance of fall armyworm in the province. We also know that fall armyworm is described in the scientific literature as a late season pest that shows preference for young late planted maize. One of the most important recommendations for smallholder farmers across the African continent where the pest is problematic is to plant early in the growing season at the onset of the first effective rains, as I noted under management strategies. Maize was planted early at the onset of the first effective rains and could explain the low number of male fall armyworm moths trapped as well. Plants were likely to have been physiologically stronger in dealing with insect pest attacks and possibly too old for successful fall armyworm larval feeding and development, as well as continued female moth egg laying. In this regard, one cannot overlook indigenous and cosmopolitan lepidopteran pests, in particular the African maize stalk borer, which is likely to be dominant in the Sidara region. Now let's quickly look at the trapping data from a farm in Richterfontein, which falls under central KZN. Okay, again, we can clearly see from the figures during both seasons that Bassiola was the threat. Clear problem on this particular farm and not the fall armyworm. Now, this is the value of keeping records on the farm, whether based on pheromone trapping or physical scouting or both, when it comes to pest management, and just the tip of the iceberg as to what can be done with such data and information. Farmers will be better able to understand pest profiles on their farms, levels of infestation, setting of economic threshold levels, timing of insecticide intervention, anticipating when in a season the pests identified are likely to become problematic based on climatic data, the effectiveness of management strategies employed in terms of crop protection products used, and so forth. Okay, so finally, to conclude, when it comes to pesticide products, users must ensure that label instructions in terms of storage, application, and disposal are being adhered to without deviation in any manner whatsoever when using a pesticide. You all would have heard me use the word instruction during discussions around the label examples. That's precisely the point. Information printed on pesticide labels are not advisories, but clear instructions. Now, other than efficacy concerns, what else compels an end user to strictly follow label instructions, particularly when it comes to dosage and application? The answer is simple. The label of a pesticide product is a legal document covered under the Fertilizers, Farm Feeds, Agricultural Remedies and Stock Remedies Act, Act number 36 of 1947. The message cannot be clearer. Do not go off-label. In addition to being effective in controlling the target pest, when used according to label and taking into account the warnings and precautions, a pesticide product will pose basically no risk to humans, animals, or the environment. I have stressed the importance of active scouting and record keeping throughout this webinar, so I'm not going to say too much more here. What I will say is that farmers need to work closely with their advisors or crop protection professionals to get the best protocols needed for managing key insect pests of crops. Extension officials or anyone advising in terms of crop protection needs to convey the importance of this aspect to farmers during training sessions, workshops, or meetings. Do not just preach the theory. As I have done today, use familiar examples to get the points across. Do not present, but rather talk to the farmers. That way, I have found to be the best in getting the message across. It is required of the farmer, especially if it is their first season of planting, to read about the subject, speak to an advisor, crop protection professional, or attend relevant courses so that the farmer is familiar with the spectrum of insects and weeds that can influence his or her maize crop at any stage. And let's not forget diseases as well. If farmers don't know what to look for, they will almost always be too late to save their crops by the time they realize there is a problem. The farmer must also know the signs of initial infections of fungus and other diseases as well. Farmers must know what part of the young growing maize plants should be inspected for damage. If they find a damaged plant, leaf, or cob and are unsure of the problem, they should keep a sample in a bag or bottle so that the pest can be identified by a professional. In some years, infestations from an unfamiliar insect can occur, catching the farmer and consultants off guard. Hence, the value of keeping records to be better prepared next time round. Remember that while scouting, farmers should also be observing crop growth and health. Pesticide application for insect pests of crops should be guided by integrated pest management principles. Misuse and indiscriminate spraying can have serious adverse effects on human health, the natural predators, and on the environment generally. Remember that fall armyworm is also known to develop resistance to insecticides as a result of indiscriminate spraying. Finally, when there are issues with control, explore all possibilities before reporting suspected resistance in the case of fall armyworm and making drastic changes. Keep calm, ask questions by considering all angles, and resolve the issue. And with that, it now brings to a close this webinar session. I trust all of you enjoyed it and also learned something. Thank you. And for those of you who may have further questions or would like to chat further around this topic, please feel free to contact me via cell or email. All the very best to everyone that will be doing the assessment. If you listened and concentrated during the training session, 100% should be the 100% should really be the order of the day. Thank you all again for being part of this webinar training session. Have a great weekend and be safe. Cheers, everyone.